This is a B4 bioenergetics video looking at photosynthesis. We've been looking at the rate of photosynthesis, and now we're looking at the kind of question where they may take this required practical and twist it into a new application. So here is a question that comes up quite often. The rate of photosynthesis in the pondweed is affected by different colours of light. So we're not doing distance of light now, we're not doing any of the other limiting factors, we are doing colours of light. Describe a method you could use to investigate this. You should include what you would measure and variables you would control. Our controlling of variables is going to give us a fair test, and what you would measure, obviously really important to show that rate of photosynthesis. So you need to include a description of how the apparatus would be used. So this is things like how you would set it up. You could talk about the fact that you're going to keep the distance of light the same. You're going to measure that distance of light using a ruler. You're going to have your pondweed, which will be producing oxygen. And then you could use filters to create the different colours of light. So maybe you could say for 10 minutes, we will have a red light filter on the beaker set at 20 centimetres from the beaker, and for that 10 minutes we will count how many bubbles of oxygen are produced, and then it will be repeated using a blue filter and then a green filter and so on. Now, one thing we should do is we should talk about the allowing the plant to kind of acclimatise to the conditions. So it's useful to say, once you've set up the filter, you allow the plant to achieve a steady rate of photosynthesis with that colour of light. Okay, so you might leave it for 5 minutes to acclimatise and then start your experiment. Okay, that will help make it a fair test. And we're using the amount of amount of oxygen produced to give us our indication of the rate of photosynthesis. We said about using it over the same length of time because we've suggested a time. We could keep the temperature the same, and quite often they talk about putting the tube that the pondweed is into a beaker, which acts as a water bath. So if you've got the hot lamp on the beaker, then the beaker absorbs some of that heat from the lamp rather than the pondweed heating up and then its temperature affecting it as well. Okay, so quite often we see that it's to control the temperature of the pondweed. Okay, and then you can do repeats, and the more repeats we do, the more accurate our mean can become, and we can take our anomalies out, work out our mean, and we get a more accurate mean. And it doesn't even hurt to explain how you calculate a mean. So you add your results together, take away any anomalies, add the results together, and then you divide it by the number of repeats that you have. I hope you found that useful. Thank you.